Hi there, my name is Marwan Sheikh, Dr. Marwan Sheikh. I am the medical director of the Assisted Fertility Program in Jacksonville, Florida. I will be discussing today the issue of prenatal genetic testing. What that means is testing the embryos for genetic problems before we put them back in the uterus in the in vitro fertilization process. The real name that we use is PGTA, prenatal genetic testing for aneuploidy. What that means is, from biology, if you remember, if you took biology in school, we have normally 46 chromosomes. The chromosomes is the DNA structure that controls everything in the body. And you know also that a male would have 46 XY chromosomes and the female will have 46 XX chromosomes. The reason this is important is because one of the problems that happens with age as we grow older is there is an increase in the number of abnormal chromosomal count. The most common of which everybody knows about is Down syndrome, where you have 3 chromosome 21s instead of 2. Prenatal genetic testing for aneuploidy would test the embryos for things like that. And it's also good for family planning by sexing the embryo. Let's say a couple have 4 girls and they want a boy. This is a good way to test the embryo to see if they have a boy embryo and transfer the boy embryo into the uterus. The other advantage of this genetic testing of the embryos is it helps us determine how to transfer only one embryo. A lot of times I have patients say, I want 2 embryos transferred or put back. I want 3 embryos because I want twins. Getting pregnant with twins puts patients at a very high risk pregnancy, risk of diabetes, gestational diabetes, high blood pressure, toxemia, etc. So we prefer to put one embryo, and checking the embryos helps us select the one normal embryo and putting it back. In general, the way to look at it, the chance of pregnancy by putting one normal embryo that has been tested with prenatal genetic testing is the same chance as putting 2 embryos that are untested. So that helps us again put one embryo instead of 2. The prenatal genetic testing tests not only the number of embryos, there are other things that it can test, which I'm not going to discuss today because it can make things a little bit complicated. But it tests mostly, as I said, for the number of the embryos. The most common chromosomes that we look at are chromosome 13, 18, 21, and the X chromosome and the Y chromosome. The X, as you know, the 2 X's make a girl, XY make a boy. So how is it done? Again, I'm not going to go through the history because it can get confusing. I'm going to tell you how we do it now. Basically, if you can see this, if we can see this, this is what is called a blastocyst. A blastocyst is a day 5 embryo or day 6 embryo sometimes. So when you get the eggs out, these are 1 cells, you fertilize them and they start growing. So by day 5 or day 6, they form what's called a blastocyst, which is about 120 to 150 cells, from one cell to about 150 cells. If you look closely at this embryo, you can find that there is a structure, lump of cells in the middle. This is what makes the baby, and there are cells around which makes the placenta. So what we do in prenatal genetic testing for aneuploidy, PGT-A, is we take about between 5 and 8 or 10 cells from this layer here and send them to a specialized lab that tests them for the chromosomes. So this raises a question: does this harm the pregnancy? There is no evidence at this stage that biopsying the embryo and taking 5 to 6 or 7 cells from from that layer, which is called the trophoblast, will harm the embryo. It does not cause any damage to the baby because the pregnancy is inside. It's called the inner cell mass. That's what makes the baby, not the placental tissue. And when we send these cells to the lab, there are different types of tests that can be done. And again, I'm not going to let you bother with the different names of these cells. The most important thing to know is the most updated testing that is done on them is called next generation sequencing. Next generation sequencing is when it gives you a much more detailed estimate of the changes in the chromosomes in these patients. Another issue that we encounter in these embryos is the issue of mosaicism. If you look at a soccer ball, you find that there are white areas and dark areas. So the surface of the soccer ball is made from two types of areas, black and white. The same thing, you could have the cells around the embryo made from normal cells and abnormal cells. And if the percentage of abnormal cells is 20% or less, we treat this embryo as normal and we still put it back. If it is more than 70% abnormal, then this is an abnormal embryo. In between, there is different discussions on how to approach that. Some people transfer them, some people don't. But definitely, if it is more than 70%, we don't transfer them back. The final thing is there any concern about this technology? We really, at this stage, do not know the long-term effect of biopsying these embryos on the children. So far, there is no evidence that it causes any problems. Thank you for your attention.